Hello everyone. Welcome to chapter 17. Chapter 17 is a pretty long chapter and we're going to spend days 15 and 16 going over it. In this chapter we're going to talk about how we take that part of the DNA that's packaged in the chromosomes called the gene and how we convert this gene to the protein. So this is ultimately getting at how do we make a functional molecule from this piece of DNA. In this first podcast we're going to cover objective 1, and this is talking about what's known as the central dogma, transcription, and how we initially go from this piece of DNA into an RNA molecule. So we're going to spend quite a bit of time going over this and give you an example. The next objective for this day 15 will talk more about the parts of the gene and how it is actually transcribed with the proteins necessary. But let's go ahead and focus here on objective 1 first. Okay, so let's talk about this thing we call the central dogma. Let's go ahead and write out central dogma here real quick. The central dogma is a concept that Francis Crick came up with. And Francis Crick is the same Francis Crick that's associated with the discovery of the structure of DNA that you may have heard about with James Watson and Rosalind Franklin. So let's talk about what this central dogma means. First, we start off with DNA. And we can draw a double helix here. And this DNA now goes to RNA. And then this RNA goes to protein. And we're going to draw RNA as a single strand here. And then protein, we're just going to draw as a globular folded up piece of protein. Now the process of going from DNA to RNA is called transcription. Okay, I barely gave myself enough room there. Sometimes in shorthand, just as I speed along in my personal notes, I'll just call transcription TXN. So if you ever see me write TXN in my notes, that's what I'm talking about is transcription. Now the process of going from RNA to protein, we call this translation. And in my shorthand, sometimes I'll just call this TLN. I'll never use these translations on an exam, but sometimes I may forget, and as I'm going through some of these podcasts, use an abbreviation. Now we're going to focus primarily on transcription today, and then translation next week. I should say next week, next class. Now let's talk a little bit in some more of a broader terms here about how this is different between bacteria and eukaryotes. And primarily I want to talk about where these events are occurring in the cells. And so let's draw a bacterium cell here. And in this bacterium, remember, all this open space here is cytoplasm. And I'm just going to abbreviate here and just write cyto. But in your notes, you may want to write cytoplasm, because that's a bacterium is just a membrane filled sac of cytoplasm. That's kind of oversimplifying it, because there's a lot of really cool things that happen in there. But what I'm trying to say is there are no organelles. And so let's go ahead and write bacteria. Now let's draw a eukaryotic cell. We know, just so we get used to the idea, that eukaryotic cells are much larger than bacteria. In fact, the size difference here isn't even well represented. The eukaryotic cell is going to be even much larger than that. We know that eukaryotes, let's go and write eukaryote. We know the eukaryotes have organelles. And I'm only going to draw one here, and that's the nucleus. That's not a very good letter there. And we write N-U-C, just for to abbreviate nucleus. And outside here, of course, is the cytoplasm. And just to be consistent, I'm just going to call this the cyto, but you should know that's the cytoplasm. We know that in bacteria, both transcription, I'm going to use my abbreviations because of space here, TXN, transcription, and translation occur in the cytoplasm. In eukaryotes, we know that transcription occurs in the nucleus, and in the cytoplasm is where translation occurs. We'll see that later on that this makes a big difference on how fast some of these genes can be turned on in bacteria compared to eukaryotes. Now let's spend a little bit more time talking about transcription. Okay, who am I fooling? We're going to spend a lot more time talking about transcription. Okay, we're going to go through transcription now. And to kick us off, we're going to draw two strands of DNA. Okay, so let's remember these are DNA. So we'll put a DNA here just so we don't forget as we start writing RNA molecules. We're also going to label the strands as being 5' here to 3'. And on the bottom it's anti-parallel, remember. So that's 3' to 5'. And these strands stay together just as a review using the complementary basis. So G's, remember, bind to C's, and A's bind to T's. Now, these two strands have different kinds of names. We're going to call them, the top one here, we're going to call the non-template strand. And the bottom one here, we're going to call the template strand. Now, for now, we're going to ignore this top strand, the non-template strand, because transcription really focuses on using the template strand, this bottom strand. And what we're really interested in knowing is what the actual nucleotides say here. So I'm going to just show this typed version of the template strand right here. So remember, what's written here in the black type is the same as here. It's the DNA as well, and it's the template strand. And now we need to transcribe this DNA molecule into RNA. And we're going to, again, do that through the process of transcription. I'm going to abbreviate this, as I warned you I might, TXN for transcription. And we have to follow the same complementary rules and the same anti-parallel rules. So let's remind ourselves what the complementary rules are for RNA. G's and C's still bind together. Guanines and cytosines still bind together. And remember, we have adenines, but we don't have thymines anymore. So the adenines, the A's, now have to bind to U's or uracils. And so what we're going to want to do is convert this to the RNA molecule. So let's just do the first one together. And what we do is we take these three bases, and we say TAC. T binds to an A binds to a U, C binds to a G. In fact, what I would do if I were you, the moment I got my exam, I would just write these down somewhere on the exam, as well as these here, just so you have them, so you don't have a mental break and forget them when it comes important, because rest assured you're going to need to know them on the exam. Okay, now this is what I want you to do, just so we can have some practice. In about 30 seconds after I explain what to do here, I want you to hit pause, and I want you to complete the rest of the RNA molecule, going through base by base below, and keep them in groups of 3, and you'll see why that's important in a moment. Also, I want you to identify if this end is the 3' or 5' end, as well as this end. Okay, go ahead and hit pause, and when you're done, hit play again. Okay, welcome back. So AGT, let's transcribe that into the RNA. A is a U, G is a C, T binds to an A. So we have U, C, A. AAA That's going to be U, U, U. GCA will be C, G, U. Because remember, there's no T's. CGG will be G, C, C. And ACT will be U, G, A. And as far as 5' and 3', we know that this end is going to be the 5' end, and this end will be the 3' end. And we know that because we have to follow that same anti-parallel rules as we did with DNA. So it's always going to be opposite what this strand is here. And we'll talk about, for the second objective, why this has to be 3' here. 3' to 5' is the template strand. Now, we want to convert this messenger RNA into a protein. How do we go about doing that? Well, we're going to do this by going through a process called translation. TLN for my shorthand for translation. And in order to accomplish this, what we need to do is look at this genetic code. This genetic code is similar to what's seen in your book. You can find it on the internet. A few interesting things about the genetic code is that it's universal. When we say it's universal, we mean that it doesn't matter if you're looking at a bacterium or a human cell, it uses this exact same code. That's pretty impressive that throughout evolution, this same code is utilized. In addition, we say that it's degenerate. And before I explain what they mean by degenerate, let me define a term that I don't think I defined, and it's a pretty important term. And that is these 3 nucleotides here, we call these codons. I'm going to write that over here, codon. Each 3 bases is called a codon. All right, so what do we mean by it being degenerate? For instance, leucine, the amino acid leucine, can be made by using one of these 6 different codons. Proline, one of 4 different. So the term they use is that the code is degenerate in that way. You'll also know by looking at the code there are 3 stop codons, UAA, UAC, and UGA, and then there is only 1 start codon, AUG. Start codon is always a methionine. All right, so how do we use this code to predict our bases? There's a couple ways you could do it. One way is you could say, okay, I have AUG. I need to figure out what the amino acid is with AUG. You could just kind of skim through this code looking for an AUG, and it's here, so you might come across it real quick. You could also do it this way, and you wouldn't require searching through each one. See, this says first base here, so we have AUG, so our first base is an A. Then our second one is a U, so we look up here, second base, put a U. And then, so we know it has to be in this column and this row right here. And then the third base is a G. So now we just follow this G all the way over here and we have AUG. And then we know our first amino acid is methionine, MET. Now what I want you to do is to once again hit pause, and then when you are done translating this messenger RNA into a protein, hit play again. Remember, what you're going to do is for each one of these codons, these 3-letter codons, use this table and determine what the amino acid is going to be. So go ahead and hit pause. Okay, welcome back. UCA. So first, remember, you see the U here, and then a C, so you know it's got to be one of these 4 here. Now we see UCA, serine. UUU, UUU, right here. That's phenylalanine, PHE. Then we have CGU. We go C, G, and so we know it's got to be one of these. And we see CGU. That's an arginine. It's my favorite amino acid. Everybody has to have a favorite one. And now we have GCC, GCC. That's alanine. ALA. for GCC. And then our last one is UGA. UGA. It's a stop codon. Now, this is a mistake a lot of people make. They want to put the word stop here. Don't put the word stop here. There's no amino acid that says stop. It just signals to the translation process to stop. So to get full credit on a question like this, you would want to have methionine, serine, phenylalanine, arginine, alanine, plus every protein, just like DNA has a 5' and a 3' end, every protein has an amino N. And you could write the word amino, or you could just write the word N, H3 here. That's an amino group. So amino, again, you could write the word amino out, A-M-I-N-O if you wanted to. And over here we have our carboxyl group. You could write carboxyl if you wanted to, but probably the best thing to do is to just go C-O-O minus. And to be technical, we should put a plus here. So that's our protein. Again, to get full credit, write the amino acid out, starting with methionine, labeling the left end. The left end is always the amino end. The right end is always the carboxyl end. Okay, that ends this objective. Go ahead and practice this again. Maybe make your own nucleotide sequence here and convert it. I'm confident there will be some kind of question on the exam like what we just went over. So good luck going through this. If you have any questions, let me know. And we'll see you in class. Bye.